Slide 1. Enzymes, Structure and Cofactors. Hello everyone, and welcome to Lecture 2 on Enzymes, Structure and Cofactors. In this video, we'll explore the fundamental building blocks of enzymes and how they function. By the end of this lecture, you should be able to describe the basic structure of enzymes, explain the role of cofactors and coenzymes, differentiate between apenzymes and holoenzymes, classify cofactors, and understand the significance of enzyme structure in determining specificity and function. Enzymes are primarily large proteins or RNA molecules characterized by complex, three-dimensional structures. The vast majority of enzymes are protein-based, and these can be further divided into two types: simple protein enzymes, composed solely of proteins, and complex protein enzymes, which consist of a protein component called the apoenzyme and a non-protein component called the cofactor. Together, the apoenzyme and cofactor form the complete functional enzyme known as the holoenzyme. Cofactors are essential for the activity of many enzymes. These are metal ions, organic molecules, or organometallic molecules that are important in catalysis. They are classified as essential ions, which include activators, metal ions, and metal-facilitated enzymes, and coenzymes, which include co-substrates and prosthetic groups. Activator ions bind reversibly to the enzyme and participate in substrate binding. When these ions bind, they induce a conformational change in the enzyme, which helps in the formation of the enzyme-substrate complex. In these cases, the metal ions do not directly participate as components of the active site. Metal ions in metalloenzymes bind tightly to the enzyme and participate directly in catalysis. Examples include magnesium, calcium, and zinc ions. These metals are often part of the active site and are involved in electron transfer, stabilizing the enzyme's conformation and bridging the enzyme with the substrate. For example, zinc is tightly bound to the active site of carbonic anhydrase, acting as a prosthetic group. Metal-activated or facilitated enzymes are stimulated by the addition or presence of metal ions. These metal ions may not directly interact with the enzyme itself. For instance, magnesium ions are required by all ATP-utilizing enzymes, such as hexokinase. Coenzymes are small organic molecules that are loosely attached to the enzyme and transport chemical groups during an enzyme-catalyzed reaction. Examples include NAD and NADP, which carry hydrogen ions, and coenzyme A, which carries acetyl groups. Coenzymes undergo chemical changes as a result of enzyme action. Coenzymes can be considered as second substrates in enzymatic reactions, primarily carrying electrons or small groups. Many coenzymes contain modified vitamins in their structures. They participate in enzymatic reactions in stoichiometric amounts and are regenerated to maintain their concentration in cells for repeated reactions. Notably, approximately 700 enzymes are known to utilize the coenzyme NADH. Here are a couple of examples of coenzymes. NAD+, or nicotinamide adenine dinucleotide, is derived from niacin, also known as vitamin B3. FAD+, or flavonoadenine dinucleotide, is derived from riboflavin, also known as vitamin B2. When a cofactor is tightly, often covalently, bound to the enzyme, it is referred to as a prosthetic group. These can be organic or inorganic in nature. Examples include pyridoxal phosphate, flavin mononucleotide, thiamine pyrophosphate, and the heme group. As we mentioned earlier, zinc in carbonic anhydrase is another example of a prosthetic group. Pyridoxal 5-phosphate, or PLP, is a prosthetic group that carries an aldehyde group, which is not found on any amino acid. This aldehyde group is essential for enzyme activity and PLP binds covalently with the chain terminal amino group of lysine at the enzyme's active site. The enzyme active site is a specific region within the enzyme structure where the substrate binds and the chemical reaction occurs. It is often located on the surface of the enzyme molecule or within a pit-like structure. The existence of the active site is due to the enzyme's three-dimensional structure. Active sites are composed of amino acids that may be distant from each other in the primary structure. They are small pockets within the larger enzyme structure and are flexible, promoting substrate binding. The active site is responsible for the enzyme's specificity for its substrate and consists of a substrate binding site and a catalytic site. Coenzymes or cofactors are often present as part of the catalytic site. There are two primary models explaining the formation of the enzyme substrate complex. The first is the lock and key model, which proposes that the active site has a rigid, inflexible shape that is an exact complement to the shape of the substrate. In this model, the substrate fits perfectly into the active site, much like a key fits into a lock. The second model is the induced fit model. In this model, the active site is flexible and has a shape that is roughly complementary to the substrate. Upon substrate binding, the active site undergoes a conformational change, adjusting its shape to fit the substrate more precisely. A good example of the induced fit model is the interaction between hexokinase and glucose. As glucose binds to hexokinase, the enzyme undergoes a conformational change to better accommodate the substrate, facilitating the reaction. In 1989, Sidney Altman and Thomas Seck were awarded the Nobel Prize for their discovery of ribozymes. Ribozymes are RNA molecules that can function as biocatalysts. Examples include 23-srRNA in ribosomes, which catalyzes peptide bond formation, and ribonuclease P, which plays a role in tRNA maturation. Compared to protein enzymes, ribozymes have a limited range of catalysis due to the limited types of nucleotides in RNA. However, both groups of enzymes use similar mechanisms of catalysis, such as acid-base catalysis and metal ions, to activate nucleophiles. Ribozymes also require secondary and tertiary structures for activity and can speed up reaction rates by factors of 10 cubed to 10 circumflex 11. Thank you for your attention. I hope this lecture has provided you with a clear understanding of enzyme structure and cofactors.